Awesome. Thank you so much. Can you hear us well? Yeah, is that
Yeah,
working?
perfect.
Amazing. Yeah, thanks a lot for the introduction and very great initiative. Also to have that a bit in Europe. I think we are lacking behind in some things. So it's very cool to see those initiatives. Yeah, and on my side, so my name is Rick, co-founder of Helical. We're going to talk a bit more about what we do. Generally speaking, in the field of biofoundation models, so DNA and RNA sequencing models. And today, really, our goal is to very briefly talk about Helical. It's not going to be the main thing, but really about those foundation models. What's the paradigm shift that comes with them? What's going to be possible in drug discovery, and why we think that everybody should be aware of them. And most importantly, if you're interested in how to use them, how you can get started in actually using them in your projects. That's really going to be the focus towards the end.
Thanks. Yeah, and then quickly to myself. So basically, I'm more from having more of an AI background. So I worked before joining or creating Helical. I joined IBM, worked as an ML engineer, and did my PhD in robotics and AI. So coming more from that side, and together with Mathieu and Rick, co-founded Helical. So I'm happy to be here.
Amazing. Yeah, so for the agenda, as I've mentioned, we're gonna be brief with an introduction of Helical so that we're aware of what we do. Then we're really gonna go into foundation models, what is out there, what exists, and finally how you can really use it. That's gonna be the main thing. So Helical was founded it's about 12 months ago, so it's quite a recent company. And we really built an open core or partially open source platform that is targeted at computational biologists and data scientists that makes it very easy to integrate DNA and RNA biofoundation models. So foundation models trained on DNA and RNA sequences and to help pharma companies to integrate those models in their drug discovery processes and get most out of those models. And today we see that there's quite little transparency around those models. So tons of models out there, new ones are coming out. It's not clear which one is the best one. And on top of that, if you want to work with those multi-billion parameter models, well, you need to write infrastructure around it, which can be quite challenging at times. And that is really the problems that we are tackling at Helical. And in a few words, where we stand ever since we have been funded. So we have raised our pre-seed or seed round, call it as you want, which allows us to really now build towards this product that I've mentioned and also serve the community. I've mentioned that partially of what we do is open source, so it's things that you guys can use also in your projects. And so we are very focused on that part. The other reason why we raised funds is that we can quickly have a great team of strong, talented people. I'm going to go into a bit more into that in a second, but yeah, that's the key things here. I think what's interesting is that we are supported by, well, in our fundraise by companies like Hugging Face or Cohere. So we come from the more AI world. And that's also a bit what we try to do. We really try to bring the best practices out of more the traditional AI space and we bring them in the bio space because we see huge potential with what's happening around those bio foundation models. But it's going to be important on how they're going to be used to get most out of them.
Yeah, so let me just quickly introduce what we think or what we talk about when we say biofoundation models, because it's there's a lot of different names running around. So I think we can call it in different terms. But basically, when I talk about biofoundation models, it comes more from a very large parameter size kind of model that has been pre-trained in an unsupervised way or self-supervised way with tons of data. So what has been the evolution a little bit of machine learning, like for more than, I guess, 50 years. So it has been very feature-centric at the very beginning. So a lot of the performances came from different models and how they can actually extract features, basically feature engineering. How could you use one model for a different task? And what were the features you had to create and mathematical formulas you had to create to get to the best results? Now, then came deep learning, which I guess kickstarted everything a little bit on the large data kind of side of things, with GPUs coming into reach for most consumers. And so basically the idea was, okay, now you don't need to care about features anymore, but you simply give the model kind of data and it learns the features by itself. And you do that for a lot of data. You will have good results in different tasks, such as image classification, speech recognition, etc. Now, in the last few years, and I guess famously with the recent research advances coming from the Transformer paper, more and more people have started doing self-supervised training on large amounts of data starting with text. So I guess GPT, BERT, and all these models, T5, have been showing what is possible with these kind of models once you start training not only on thousands of text documents, but millions and billions. And in an unsupervised way, so really you don't have to care about labeling it yourself, but you would let the algorithm learn by itself how to go through that data. And this is really, I think, one key aspect of biofoundation models is the aspect that they are self-supervisedly pre-trained on large amount of data, and then can be reused. One single model can be reused in different tasks to actually accommodate for different needs. And so just to very briefly explain what I just said, maybe some images and also coming from language. So really these large foundation models serve as a basis for later adaptations or adjustments you want to do for your tasks, which we call or through fine-tuning or probing. So what this means is basically you can extract features from your documents or modalities, so text, for example, or DNA, and basically use that representation with a logistic regression, for example, an SVM, or other kinds of models to actually then do downstream tasks. And the key characteristics of these models is they are pre-trained, so basically a large amount of the computational costs have been done upfront if you reuse the pre-trained model. And the fine-tuning cost is actually marginal in comparison to that. But what is neat about it is that the fine-tuned model will be much more powerful than if you would actually just use a new model from scratch, because it has seen so much data during its pre-training process. And that speaks to its versatility, really, that you can use it in different contexts with the same basis, which we think is really one of the key aspects as well of why this is interesting in biology. And then the scalability, there's something called a bitter lesson, and we'll go into that. But basically, it seems that the more data you throw at it and the more compute, the more kind of challenging tasks you can start to handle. And we've seen this trend of increasing parameters and immense sizes you just grow increasingly or continuously, which obviously brings some other challenges, such as infrastructure challenges, data engineering challenges, etc., etc. So that's really, I think, what we think when we say foundation models. And so in biology, we've seen a bunch of them, but we will talk about two types of them at the moment. So basically, we have DNA sequence models that really look like the nucleotide transformer, hyena DNA, EVO. Multiple research groups have published different models. The idea is always a little bit the same. You have, similar to text, you have sequences of nucleotides, and you try to predict what is the missing nucleotide or what is the next coming nucleotide. And basically, you do that on thousands of genomes. So you don't have to label it. So you just shuffle out some nucleotides and try to predict which nucleotide is actually missing. And the nice thing about that is you can do it at scale, because now you can really use this. On the other hand, there's something also new models that have come into play here. So single cell expression models. So basically, they are trained or similarly pre-trained in a sense to large parameter size and data sets. So in an unsupervised or self-supervised way. But on the other hand, they don't really they don't look at sequences, but it's more on the expression data. So you try to actually extract the patterns that you have in terms of relationships between different expression data. And so, again, these different types of models have been pre-trained on either thousands of genomes or millions of cells. So the latest one has been pre-trained on 105 millions of cells, which is the gene former V2, which makes it very...
I think you went on mute.
We can use these pre-trained models on different tasks. Yeah, sorry, I just bumped against the button. So we think this is kind of a paradigm shift because, one, you can now just feed it the large amounts of data that become available. So we see more and more DNA sequences becoming available. GWAS studies, the price of DNA sequencing goes down. More databases actually release public data sets. SEDEX gene census database is huge. More and more studies come out. So this is actually ideal because at the beginning, you don't know really how to leverage this data, but with these self-supervised versions of data, of modeling and pre-training, you can actually do this, and you can leverage every single data set here. Of course, you need the right resources and infrastructure, but technically speaking, it's feasible for the model to actually extract knowledge there, which is very interesting because it has never been able or could never have been done before. And then there's an economical reason, right? You once trained, the costs are marginal to adapt it. And it doesn't mean that the model is the best model for every single task, but it means that basically the knowledge it has learned during that pre-training phase actually can be reused across your tasks, across your groups. You can leverage it for multiple experiments, and you simply need to adjust it with fine-tuning. And finally, a new way of actually creating workflows, I think, will come up because now these models cannot be used only as prediction models or regressor models, but basically you could try and actually leverage them as kind of simulators or pattern extractors for your models or for your data, sorry, and then you could use that representation or embedding in a different use case that you're just working on or in a database to do similarity searches, semantic searches, and all of that requires a little bit of rethinking about these workflows with that biofoundation model at the center to leverage these new capabilities of latent representation of complex patterns that you can extract. So we think there's new opportunities here and new ways of thinking about how we can create value with them and with different machine learning workflows, which differ a little bit from what we've seen previously. And then also what comes with that is this beta lesson that I was talking about before, where we can clearly see that the parameter amount of parameters that we see in models is actually increasing drastically across language models. So in green on the left side, and on the right side in purple, this is for DNA sequences. We see the same evolution happening on the DNA sequences, even though it's not that clear yet that this trend will continue. But we're already talking about large amounts of data and large amounts of parameters that you need to distribute. You need big HPC clusters to train them on. And so Evo and nucleotide transformers both have used a lot of GPU power to actually just get there. But now they are available, and you can use them for your research or for your experiments, even though not all of them are open source. They don't have like a non-commercial license. Then the big missing piece of this, coming with new technologies or new kind of, let's say, algorithms, there's like a big need of having good benchmarking framework that actually shows us, okay, well, in these kind of scenarios, especially in biology, traditional methods will outperform the biofoundation models, and in others, biofoundation models might be better, and which one is better on what task. Now, there isn't really a structured way of doing this at the moment. So at Helical, one part of our open source program that we're doing and contributions that we start doing is basically building out a framework with clear data sets that actually allow us to benchmark each single biofoundation model as well as other traditional methods to actually go and look for the results in each task. It can be integration tasks, it can be self-annotation tasks, it can be a lot of different kind of tasks. So really, we try to have like a structured way of doing this, and I think this is also a call out or shout out to everybody who's interested in contributing to these efforts, because we think this is a community effort in terms of getting the best ways of actually benchmarking these kind of models, not just for your work, but basically for every work that is done, so that nobody can say, oh, well, this is the best model. And we use this because we think it's the best, because if you test it on different tasks, then it will outperform, or will be outperformed by simple methods. So we think this is super important. And we will show you a little bit how you can contribute to that as well later on. So let Rick continue with how you can get started on that part. Oh yeah, there's a question in the chat. Sorry, do you want to do it immediately, or should we do that in the Q&A session?
We can wait till the end. You guys can.
Okay. Anyway, it's gonna be quick from here. The question is now, indeed, how you can get started. I think it's quite simple. A part of our technology is being open sourced, especially our model library. This means that basically on our GitHub that you can see here, maybe you can also post a link in the chat to make it a bit easier. you will find all of the recent biofoundation models on DNA, trained on DNA and RNA data in there, meaning that it's very easy to use them. We have functionalities such as fine-tuning that's already included in there. And I think most interestingly, we have example notebooks that should make it very easy to get started on use cases that are quite common for the space of bio, such as cell type annotation, which is, I guess, still somewhat of an unsolved problem, generally speaking. However, we have already included the workflows on how you can leverage those foundation models in a very easy way on cell type annotation and also on fine-tuning scripts that you can use directly from our GitHub. So that will make your life as easy as possible. In case you use it, don't forget to give us a small GitHub star. Of course, the effort we put in the community, it's always great for us to see if it's also growing. So feel free to leave that, and also feel free to give feedback on it. I mean, if you see things that are not great, we are more than happy to look into that and adjust it. And maybe on the last note, as I mentioned in the beginning, we have raised first funding round, and we currently already have an amazing team. You see that we have mainly machine learning engineers, computational biologists, and we are now expanding that team with 2 open positions. So on the software developer side and computational biology side, especially with ML experience. So in case that sounds interesting to you guys, then feel free to reach out or feel free to apply directly. And we are more than happy to have a chat about what these positions could look like.